Okay, welcome to today's live, everyone, which is going to be about digestion and absorption. So we're going to be going through some exam questions on digestion and absorption, but also we'll have a look at some of the activities in the active recall workbooks, so you can just see how the information that you learn in your lessons could come up in the exam questions, but this is just a way to have a look at it, understand it a bit better before jumping in. So that's actually what I'm going to have a look at first. So I know one of the first questions, that was actually about starch. That wasn't my first question though. Here it is. So this one's to do with lipids. So maybe let's start on the lipids bit. Here we go. Which is often a topic that people find really hard, and it's one of my most watched videos on YouTube, which again shows me how hard people find it, because I can always tell based on which videos are the most watched. That's clearly the ones that people find the hardest because they're going for extra help. So let's have a look first of all at what are micelles. So micelles, what would we say is a micelle? TikTok, this is for AQA, but there's lots of overlaps with OCR and all the other exam boards. So micelles, the first thing that I would say is they are water soluble. So water soluble vesicles, and they are formed of fatty acids, glycerol, you also get some monoglycerides, and bile salts. So that is what our micelle is. The next part of this question though is then saying, just gonna try and shift this over a bit to work out which way I need to move it for Instagram to see better. I feel like it needs to be this way, but I'll suddenly see if it's about to make it a lot worse. And let me just check on here. Yeah, I think that's probably okay. Next question then we have is, why can the contents of a micelle move by simple diffusion across the membrane? So the micelle itself doesn't transport across, it releases the contents at the membrane, and then what diffuses across is the fatty acids, monoglycerides, which are lipid soluble. So that's what we're going to have here, is the fatty acids and monoglycerides are non-polar, or in other words, that means they are lipid soluble, so can dissolve in that phospholipid bilayer. And that's why they can then move across by simple diffusion. So next question is, what happens to the fatty acids and monoglycerides once they've been absorbed into the cells? So this is now going on from digestion, it's now going on to absorption, but then also what happens after absorption. So once they're in the cell, once in the cell, these are modified back into triglycerides. And that's done inside of the Golgi, that should be capital, Golgi body, and also the endoplasmic reticulum. Right, final activity that we're going to have a look at on the lipids before we go on to some exam questions is this diagram here. We need to have a go at labelling it. And the diagram, it tells us, label the diagram to describe lipid absorption. So that's what we're going through, lipid absorption. So we need to have a look at those different stages. So this first picture then is pointing at this circle. Those are the micelles. So then it's saying, well, what is this? So the micelles would have delivered these fatty acids and monoglycerides, and then it releases it at this point at that cell surface membrane, so it can then diffuse across that cell surface membrane. So that's what I'm going to say here. Fatty acids and monoglycerides leave the micelles and enter, and really I should say diffuse there, so I'm going to say enter, so diffuse into the epithelial cell. There we go. Next then we have this image here, and at this point this is where the fatty acids are going to enter that Golgi body to reform into the triglycerides, just like we said on the previous page. So fatty acids enter Golgi to form triglycerides. Now at this point you could also have proteins being added and other modifications which would make it a chylomicron, which I'm just going to write here. And those will then, so that's what we've got here. We've got those globules, so fatty globules combining with proteins. So that is when it combines with protein. And that is then going to go on to form a Golgi vesicle. Not form, but it's released via the Golgi vesicle. And we can then see at this point that Golgi vesicle is fused with the cell surface membrane again, and we have this exocytosis to release the contents of that vesicle to the outside. So we've got exocytosis, so vesicle fused with membrane, and contents released. For OCR, you actually have to need to know more details about exocytosis. For AQA, you don't cover exocytosis. Same with Cambridge International and EdXL, you learn more about exocytosis. And then the final step we've got here is it enters the lacteal and is transported in the lymph. And there we go. That is the absorption of the digested lipids. So then if we have a look at what the exam question was, because as I said, that's like the point of this active recall workbook. It's bridging the gap between learning the information and checking it, and you do get the answers with the booklet as well. And then it feeds into, you can see how what you've just written down might link to an exam question. So let's take a look at this example of an exam question for 5 marks. So we have to describe, so that's the command word here. Oh, I want that a bit neater. There we go. Describe the steps involved in the absorption. So we're looking at the absorption and transport of digested lipid molecules from the ileum into the lymphatic vessels. So it's essentially what we have just said, but we need to put that as 5 key bullet points for the key marking points. So we now need to think about what we just wrote down and turn that into a 5-mark answer. So that is what we're going to have a go at. Let's see, I'm going to have a little look in the chat box, what sorts of things we've got. This is A-level, so there's no higher or lower tier, it's not GCSE. So this is A-level AQA biology, but it is still relevant for multiple exam boards. But this specifically is a question based on AQA. So the first thing then that we talked about on the other page was those micelles. So I'm going to say micelles contain bile salts and fatty acids. You could say monoglycerides as well. Oops, I just realised I completely knocked that out. So there we go. And monoglycerides. The reason I bring that up is in terms of how that links to the digestion and then the absorption is we need to know that the fatty acids can then move towards the cell surface membrane. So I'm going to say that makes the fatty acids soluble. And by that we mean in water, because the cytoplasm is a water-based solution. So if they're now soluble in water, they're soluble in the cytoplasm and they can move along. The session will be about another 20 minutes. I just saw someone ask that. So it's normally about 20 to 30 minutes long. We'll be going for 10 minutes already. So that's how long it will be. Right, where was I? Looking at these exam questions. So that then means, how is it absorbed? Fatty acids diffuse into the epithelial cell. And because they said from the ileum to the lymphatic vessels, I'm going to point out it diffuses into the epithelial cell from the ileum. So we've now got our start point. So now we've got this bit. We've talked about how it goes from the ileum into the epithelial cell, but then we need to know what happens in the epithelial cell and then how it gets to the lymphatic vessel. And that's where we then said that triglycerides reform or form in the Golgi. And then vesicles containing the triglyceride, or it could be a chylomicron, but I'm just going to stick to triglyceride here, fuse with membrane. I'm actually going to say move and fuse with membrane, which I'm going to just put in brackets, exocytosis. And then the last thing then is say, well, how does that get it into the lymphatic vessel? So I'm going to say it's released and enters the lymphatic vessel. There we go. So that would be 1, 2, 3, 4, 5, 6 bullet points for a 5 marker. So yeah, we've definitely covered our back to make sure we've covered all of the key concepts there. So let's have a look at what the next question is. Describe the stages of protein digestion in humans, including the enzymes involved in their role. So it's 4 marks. Let's just go back to the active recall workbook and see what we had there to guide us through this before doing an exam question. So in the active recall workbook, it points out the 3 enzymes where you've got to then describe the role. And then it goes into a bit more. We've actually done the lipids bit already, the 2 molecules needed. That was bile salts. And we did actually talk about lipase, but the lipase enzyme. But I'm not going to do this bit because we've just done lipids. I'm just going to focus on this bit up here. So into the chat box, let's see what we know. So describe the roles of, we have endopeptidases, exopeptidases, and then membrane-bound dipeptidases. So those are our 3 options. Let's see, do we know what each of these do? And I can actually see in the chat box, I've got quite a lot of contributions coming in from Instagram. That's excellent. TikTok, I can see some coming in. Yep. Someone said, when do the lives start? They start 8 p.m. every Thursday, and they just automatically start on Instagram, TikTok, and YouTube simultaneously. So you just have to find me on one of those. And it is AQA A-level biology. Right, so endopeptidases. I can see some answers are coming in already. Excellent. So for our endopeptidases, these will hydrolyze peptide bonds. Now that's actually the starting point for all three of these, but they all hydrolyze peptide bonds in different places. So endo means inside. So if it's an endopeptidase, that means it's an enzyme that hydrolyzes peptide bonds from within the middle of the polymer chain. So I'm going to say hydrolyzes peptide bonds between amino acids in the middle of polymer chain. Exo means outside, so that means it's going to be on the end. So again, hydrolyze peptide bonds. This time it's between amino acids at the end of polymer chain. And then the last one are dipeptidases. Again, it hydrolyzes peptide bonds. But this time it's between two amino acids. So if you just had two now, so you started off with your long polymer chain, the endopeptidases, those are going to hydrolyze the bonds in the middle, and so I'm just going to color code that there. The exopeptidases will hydrolyze any at the end, and then if you do have any two that are left, that's what these membrane-bound dipeptidases can do. So hydrolyze peptide bonds between two amino acids. Okay, there we go. So where does digestion occur? I saw lots of these answers coming in. So it starts in the stomach, and then actually continues. This doesn't all happen in the stomach, so it continues in the small intestines, but you need to be specific at A level and say which part. So in the duodenum to start with, that's the top end of the small intestines, and then down into the rest of the small intestines, which is the ileum. So continues in duodenum and is fully digested in the ileum, because that's where you have those membrane-bound dipeptidases. Hopefully you can read my scrawling, because whenever I'm reading and writing at the same time, this is what you get, an absolute scribble. Right, let's go back then to what the exam question was. Now we've just done a little recap. So you had to describe the stages of protein digestion in humans, including the enzymes and their roles. So it's 4 marks. It's pretty much going to be what we've just gone through in that exam question. So the first thing is, we said for all of them, there is hydrolysis of, hydrolyze those peptide bonds. So that's my first bit. Hydrolyze peptide bonds between amino acids. Then the other marks are going to be for talking about all 3 enzymes. So we've got endo, exo, and di. And then I'm just going to add in peptidases. So I'm going to say breaks polypeptide into smaller chains. I'm actually going to add a bit more here to what we said. So hydrolyzes peptide bonds in middle of polymer. This one here is going to break off terminal, which means at the end, amino acids, because, or you could say hydrolyzes the peptide bonds, we kind of covered that up there. And then the dipeptidases would be, it will hydrolyze the dipeptides into amino acids. Then we've got 4 bullet points plus my extra one down there just to cover those 4 key marking points. So next then, our next question, describe how starch is completely broken down in humans, including the role of the enzymes of the digestive system. So this now is exactly the same question that we just had, but this time it's going to be for starch specifically. So let's just see if I had a start point on this. Yeah, so here's our start point, just to link this back to the active recall workbook, hopefully to just help a little bit before we go into that exam question. So I'm going to skip this one here and just go on to the carbohydrate specific ones. We might need to know this bit, so let's just add this in. So the 3 locations that digestion of carbohydrates happens is, it starts in the mouth, then it happens in the duodenum, which is the top part of the small intestines, and then it's completed in the ileum, which is the lower part of the small intestines. The enzymes which are involved in this then, we have amylases, and the location of those being produced would be in the pancreas and the salivary glands. So because it's produced in the salivary glands, that's why carbohydrates can be digested in the mouth. Then anything that is digested there is swallowed, goes down the esophagus into the stomach, and then out of the stomach into the duodenum. And at that point, the pancreas will be releasing in amylases into the duodenum, and that's why there's digestion there. And then finally in the ileum, that's where we get to the second stage, which is the membrane-bound disaccharidases. So similar to the concept of the proteins, which also had membrane-bound, but those were dipeptidases, you also have membrane-bound disaccharidases in the ileum. And these are all of the enzymes that are embedded within the walls of the ileum. But I've realised I haven't actually done the products bit here. So that would be polysaccharides would be hydrolyzed into disaccharides. So for example, maltose. So then here, this would be our membrane bound disaccharidases. These are in the ileum, I'm just going to say small intestines as well, just to cover that. And this is where you would have maltase, sucrase and lactase. Those 3 enzymes can convert maltose, sucrose and lactase. We've got some synoptic link here into biological molecules. All of them would produce glucose, but then sucrose, you'd also have fructose, and lactose, you'd also have galactose. But basically, it'd be monosaccharides. So let's see how that would potentially translate to this 5-mark question, which is describe how starch is digested. So we need to know, how is starch digested? So describe a starch completely digested. Yeah, so is that not absorption? So for this one, it would be linked to all the key things that we just said. So I'm going to say amylases produced by salivary glands. I'm going to say hydrolyze polysaccharides. So because I've said amylases, I'm just going to add in for more detail. For example, starch into disaccharides, which could be maltose. I'm going to also add in pancreas releases amylases into duodenum for further hydrolysis. Then I'm going to say membrane bound disaccharidases, for example, maltase, and that will hydrolyze maltose into glucose, which is a monosaccharide. Right, we've got 1, 2, 3, 4, 5. I think that covers everything. Let me just check if anyone's added anything extra into the chat box. And I don't think so. I think those are all the key points. So that takes us to one of the final questions. I think we've covered most of what I want to do, just to do an overview of digestion and absorption. So we've covered starch, proteins, and lipids. I've done more actually on the absorption of lipids and more on the digestion of these two. I haven't done the absorption of all of them, but we are at 1:30 now, so I'm going to stop it there. But I'll have a little look to see if there are any questions coming up in the chat box. I'll just leave it on this question in case people were trying to look at those. So I'm going to start on YouTube. Only question I had on YouTube were, is this A-level or GCSE? It's AQA A-level to answer that question. On Instagram, I don't think we've got any new questions coming in at the moment. TikTok, I think there were a few. Let me just see. Do you think the Oxford A-level textbook, watching your videos and practice questions is enough? The OUP textbook is good. It does go into more detail in some places, but that's my favourite of the textbooks. Watching my videos would be the same really as the textbook. Both of those are passive because it's just taking in the key information. And then exam questions. I think you're missing a middle step there. And if you did come to my free webinar where I talked about revision, you need to have understanding, then you go on to remembering, then you go on to practice questions. And the understanding stage would be when you might use the textbook or my videos to understand the information, but then you need to make sure you can remember it. And that's when you need to do things like this, the active recall workbook, where it's testing, did you understand, do you remember it? Because can you answer these short answer questions and do these like little labeling activities where you can use your notes to help you. Or you might be using flashcards to test, can you remember the key information? But then once you've got to that stage, then you go on to the exam questions. So that's what I was trying to model here, doing the remembering part first to make sure you can remember the key information, then do exam questions. Because otherwise sometimes if you jump straight into exam questions and you can't remember the information, then it can be really demoralizing, demotivating, because you end up not remembering it, you don't get the marks, and you haven't really practiced your exam technique. So those are 3 steps to follow: understanding with notes and videos, remembering with active recall, such as my workbook or flashcards, or testing each other, and then do exam questions to see, can you then apply all of that to an exam question. So I hope that helps. And that is it for today, everyone. I will be uploading all of these as little reels and TikToks, and you'll see the full length one saved on the YouTube channel as well. But that's it for today. If you do have any topic requests, just send them over to me. But for now, I'm going to leave it there, and I will see you next week, everyone.